Hi, welcome to my Chapter 8 presentation. We are going to talk about the nervous system. And the nervous system exists kind of for like three broad functions. It's going to monitor the body's internal and external environments. It's going to integrate sensory information, and it coordinates voluntary and involuntary responses of many other organ systems. So to start off, the nervous system is comprised of two separate parts. It's comprised of the central nervous system and the peripheral nervous system, CNS and PNS for short. The central nervous system is comprised of the brain and spinal cord, while the peripheral nervous system is comprised of nervous tissue outside of the CNS. And so the CNS's main job is receiving, processing, and responding to sensory information, while the PNS is going to be like communication connection between the CNS and the rest of the body. And the PNS is going to have two different divisions. It's going to have the efferent division, which brings information to the CNS from the receptors, and the efferent division, which is going to carry motor commands from the CNS to peripheral tissues and the systems. On top of that, the efferent division is comprised of two different nervous systems. That's the somatic nervous system and the autonomic nervous system. So the CNS and the ANS, a lot of NS acronyms. So the somatic nervous system is going to be comprised of the skeletal muscle effectors, and effectors are like the target organs and tissues whose activities change in response to neuromotor commands. And the autonomic nervous system is going to have, again, two separate divisions, the parasympathetic division and the sympathetic division. And the parasympathetic division is going to be comprised of smooth muscle, cardiac muscle, and glands, while the sympathetic division is all of those three plus any adipose tissue. So going back to the efferent division, afferent, I'm so sorry. My E's and my A's, I get them mixed up. My bad. But the afferent division is going to be comprised of two receptors, the somatic sensory receptors and the visceral sensory receptors. And the somatic sensory receptors are going to monitor the outside world and our position in it, while the visceral sensory receptors monitor the internal conditions and the status of other organ systems. Woo! And so how the PNS is made up of nervous tissue outside the CNS. Nervous tissue is going to be made up of neurons and neuroglia. And neurons are the basic functional units of the nervous system, and neural functions involve the communication of neurons with one another and with other cells. If we look right here, we kind of have like the basic structure of neurons. We have the mitochondria, we have the nucleus, the nucleolus, and the Golgi apparatus. We've talked about neurons before multiple times. The axons, the collateral, the axon terminals, the cell body, the dendrites, the nerve cell body. All of these things that we've talked about before, once again, they're going to be in most organ systems. All right. So the neuroglia in the PNS are going to be comprised of two different types of cells, satellite cells and Schwann cells. And satellite cells are going to surround and support neuron cell bodies in the peripheral nervous system, while Schwann cells cover every axon outside the CNS. And we have some type of neuroglia in the CNS because this was neuroglia in the peripheral nervous system, but neuroglia in the central nervous system are going to be ependymal cells, and ependymal cells are simple cubital epithelial cells that line fluid-filled passageways within the brain and spinal cord. Microglial cells are microglia that phagocytes that move through nervous tissue, removing unwanted substances. Astrocytes are star-shaped cells with projections that anchor to capillaries. They form the blood-brain barrier, which isolates the CNS from the general circulation. And then I'm going to call these oglio, because I guess I could just ogliodendrocytes. I feel like I'm saying that right. I'm going to say the whole word. I'm going to say the whole word. Oligodendrocytes, I had some confidence, and the confidence went out through the window. I am so sorry. Oligodendrocytes are cells within the sheet-like processes that wrap around the axons. And with these oligodendrocytes, the small gaps between adjacent cell processes are going to be called the nodes. And these are the nodes of Ranvier. All right. So within the peripheral nervous system and the central nervous system are going to be called, there are these called gray matter and white matter. And the gray matter and the PNS are going to be like ganglia. These are like connections of the neuron cell bodies in the PNS, while the white matter are going to be like the nerves, so the bundles of the axons in the PNS. In the CNS, gray matter is going to be the neural cortex, the nuclei, and the white matter is going to be the tracts and the columns. I left out for gray matter the centers and the high centers. But gray matter on the surface of the brain is going to be the neural cortex. The nuclei are going to be collections of the neuron cell bodies into the interior of the CNS. Centers are going to be the collections of neuron cell bodies in the CNS. Each center has a specific processing function. And higher centers will be the most complex centers of the brain, while with white matter, tracts are going to be bundles of CNS axons that share a common origin, destination, and function, and columns are just several tracts that form an autonomically distinct mass. Right. So pathway, and we see right here in the diagram that receptors, they receive the information, they're going to go inside, get the process, and then the effectors right there. All right. So pathways are going to be centers and tracts that connect the brain and other organs and systems in the body. The ascending is going to be the sensory pathway, while the descending is the motor pathway. All right. So here we have a little diagram of resting membrane potential of a neuron. And the membrane potential of like an unstimulated resting cell is known as like the resting membrane potential. And the resting membrane potential of a neuron is going to be about negative 70 MV. The minus sign indicates that, like the cytoplasmic surface of the plasma membrane contains an excess of negative charges compared to the extracellular surface, which is kind of almost not the direct opposite of action potential, but kind of. So if we go right here to this like little portion of our diagram, action potentials are going to be our propagated changes in the membrane potential that, once started, affect the entire excitable membrane of the axon. Action potentials depend on the presence of voltage gated sodium and potassium ion channels. And these voltage gated channels, they open or close in response to changes in the membrane's potential. So action potential, they're gonna become open, and when they're resting, they're gonna become closed. And just steps in the generation of like the axon potential. The first step is gonna be like graded depolarization. It's gonna be caused by an opening of the chemically gated sodium ion channels, and it's usually like on the axon hillock. So right here we have the depolarization of the threshold, leading into the action of the sodium ion channels and rapid depolarization. If we see right here, we can see, kind of like charges. We're going to go, and then it's going to go into the inactivation of the sodium ion channels and the activation of the potassium ion channels, while closing of the potassium ion channels, and then it's going to return to the resting membrane potential. So it's a cycle, resting into action, into resting into action. So it's all together. And if I didn't do it before, when it says these propagated changes, propagation is just like the process by which a nerve impulse or like an action potential travels along a neuron. Yes, just in case I didn't mention that earlier. I feel like I didn't. All right. So we go right here talking about propagation. We have continuous propagation and we have saltatory propagation. So basically, in unmyelinated axons, an action potential moves along by continuous propagation. The action potential spreads by depolarizing the adjacent region of the axon membrane, and this process continues to spread as a chain down the axon. While in saltatory propagation, because myelin limits the movement of ions across the axon membrane, the action potential must jump from node to node during propagation. This results in much faster propagation along the axon. No. There we go. So yes, so that one's just gonna be a little more faster because like the myelin limits the movement, whereas in the one that doesn't have any myelin in it, it's just continuous propagation. Right. And so here we have the structure of the synapse. I feel like I'm saying that right. Yes, the synapse. So right here, it looks very, there are some little structures in here that are going to look some similar. So like the mitochondrion looks similar. The synaptic vessels are going to look a little similar. But we have something new. We have the axon of the presynaptic cell right here, or axon isn't new. I don't know. But I guess like talking about like the structure of the synapse is new. Axon of the presynaptic cell is going to come right here. Do you see the edge? The axon terminal surrounding it, the presynaptic membrane, also, like surrounding it on the inside. The postsynaptic membrane, the one that's like surrounding it on the very edge. And then the synaptic cleft, which kind of like, what's it called? Well, not combines. Attaches the postsynaptic membrane and the presynaptic membrane together. So pre, before, post, after. That's how I remember it. All right. So here we have oh yes, the what's it called? The chemical synapse. Yes. So first we're going to look at like the steps, kind of like how the chemical synapse happens. So first an action potential arrives and depolarizes the axon terminal. Then like the extracellular CA2 is going to enter the axon terminal and it's going to trigger the exocytosis of the ACH. The ACH binds to the ACH receptors, resulting in depolarization of the postsynaptic membrane, and the depolarization ends as ACH is broken down by ACHE. All right. Ooh, so here we have a diagram of the different sections of the brain. We have the cerebrum. We see right here, the two, basically like the two halves of the brain. We're going to have the frontal lobe, or like, the left cerebral hemisphere. And we're going to have the parietal lobe. We're going to have, and this on the outside, right here, we're going to have the diencephalon and the brainstem. Yes, they're all connected. All right. So the frontal lobe, as we see right here, is this like lighter pink section over here. It's going to have the primary motor cortex. It's going to have the premotor cortex, and it's going to have the prefrontal cortex. While the parietal lobe is going to have the primary somatosensory cortex and the somatosensory association area. The occipital lobe is going to have the visual association area and the visual cortex, while the temporal lobe is going to have the auditory association area, the auditory cortex, and the olfactory cortex. All right, and if this is just kind of a recap of some words to make sure that we know them. The receptor, there are sensory structures that detect changes in the environment or respond to specific stimuli. Nerves are going to be bundles of axons, and ganglion are going to contain the cell bodies of sensory neurons. If we come over here, we have some classifications as to where we're going to have certain nerves. So when it comes to C1 through C8 on our spinal cord, those are the cervical spinal nerves. T1 through T12 are going to be the thoracic spinal nerves. L1 through L5 are going to be the lumbar spinal nerves, and sacral spinal nerves are going to be S1 through S5. And at the very bottom, the CL1 is going to be the cycloglial nerve. And we kind of have our little, and these are the vertebrae, by the way, and we have a little diagram of the vertebrae inside. So we have the anterior root, this kind of like yellow part at the very bottom, and the spinal nerve that's going to connect to it, up into the spinal ganglion, up into the posterior root that actually connects to the vertebrae. And in the vertebrae, we're going to have white matter kind of surrounding it and gray matter on the inside, while the central canal is going to be in the very middle. All right. So cranial nerves, we have cranial nerves 1 through 12. The olfactory bulb is going to be cranial nerve 1. It's for special sensory. While the optic nerve, our nerve 2, is going to be also special special sensory. It's going to be the retina of the eye. Oh, I didn't mention this for the other one, but the olfactory bulb is gonna have olfactory epithelium, while the optic nerve is gonna be in the retina of the eye. The oculomotor nerve 3 is gonna be for motor functions, and sorry, I had a yawn there for a second. The oculomotor, which is our third nerve, or nerve 3, is going to be for motor functions, and for the innervation, it's going to be inferior, medial, superior rectus, inferior oblique, and intrinsic muscles of the eye. The trochlear, which is nerve 4, is also going to be for motor functions. It's superior oblique muscle of the eye. The trigeminal, our nerve 5, is going to have like a mix of functions. For sensory, it's going to be like orbital structures. It's going to be the nasal cavity, skin of the forehead, the eyelids, eyebrows, nose, act. While for the motor function, it's going to be like the chewing muscles. Abducens, which is our nerve number 6, is going to be for motor functions. It's the lateral rectus muscle of the eye. For facial, nerve 7 is also going to be a mixed functions. For sensory, it's going to be like our taste receptors. And for motor, it's going to be the muscles of our facial expressions, our lacrimal glands, and our submandibular and subguineal salivary glands. I have been trying to pronounce this for like the past hour, and I have not been able to, but I'm gonna try my best. The vestibulocochlear special is going to be for special sensory functions, as the cochlea and the vestibuli. The glossopharyngeal is going to be for mixed functions. For sensory, it's going to be the posterior 1/3 of the tongue. It's going to be the pharynx and the palate. It's going to be receptors for blood pressure, pH, oxygen, and carbon dioxide. The vagus, which is our nerve 10, is going to be mixed functions as well. For sensory, it's going to be the pharynx, the external acoustic meatus, it's portion of the external ear, the diaphragm, the visceral organs in the thoracic and abdominopelvic cavities. For motor, it's going to be the palatal and the pharyngeal muscles and the visceral organs in the thoracic and abdominopelvic cavities. For accessory, we're going into nerve 11, is for motor functions. It's going to be voluntary muscles of the palate, pharynx, and larynx. And for the hypoglossal, which is our last nerve, nerve 12, motor, is going to be the tongue muscles. And obviously these are not our only nerves, but these are our cranial nerves. All right, and then to also just give some more definitions, preganglionic neurons are going to be the ANS motor neurons in the CNS, and they send their axons called preganglionic fibers to the autonomic ganglia outside the CNS. The ganglion is a collection of nerve cell bodies outside the CNS, and the postganglion fibers are going to be axons of the ganglionic neurons. They're going to leave the ganglia and innervate cardiac muscles, smooth muscle glands, and in some cases, fat cells. All right, looking at here is where did I had my titles. Where did it go? No, my titles. Anyways, but this is gonna be, I believe, did I mix up my slides? I did not mix them up. Okay, so at least they're in order. So this is gonna be the sympathetic and the parasympathetic divisions of the ANS. And this is just going to have, like, the different structures of it, like, the different, like, parts. So, for this one is going to have the T1 through L2 vertebrae, while the parasympathetic is going to have, the pons, the medulla, this bottom part of the spinal cord that leads to S2 to S4. It's going to have some pelvic nerves that lead into that. Yes. But this is the diagram that I use whenever I want to look at the differences. They do Obviously, there are some similarities because we see that some of them, you know, both of them for like the eyes, the salivary glands, the heart, and the lungs. But on the one side, on the left, we see that they have the tear glands, they have the pons, they have the medulla, they have this bottom part of the spinal cord for S2 through S4. And then on the left side, we see that all not all the other vertebrae, but T1 through L2, we see that those vertebrae are part of it. We see that you know, over here, we have the large intestine, small intestine, but on the other side, they have the rectum as well. On the left side, they do not have the rectum. On the left side, they have the adrenal medulla, and the adrenal medulla is going to be like the center of each adrenal gland, and it's like a modified sympathetic ganglion. Its ganglionic neurons are gonna have very short axons, and yes, I think that's gonna be it for that. But here we just have some, what's it called, structures, the sympathetic effects and the parasympathetic effects. So when it comes to the eye, tear glands, there are no sympathetic effects, but for the parasympathetic effects, it's going to be secretion. You know, for like blood vessels, sympathetic effects are going to be like vasoconstriction and vasodilation, but there's going to be no parasympathetic effects. In the heart, sympathetic effects are like increased heart rate, force of contraction, and blood pressure, but parasympathetic effects are decreases in heart rate and forces of contraction and blood pressure. You know, so there's just different. Some of them have very similar effects, and then some of them will have effects for one and not for the other. Like for the skin, the sweat glands, there's no parasympathetic effects. For the aerator, erector pili muscles, there's no parasympathetic effects. But for sweat glands, sympathetic effects are increases secretion. For the aerator pili muscles, it's going to be the contraction and erection of hairs. For the adrenal glands, sympathetic effects are going to be secretion of the epinephrine and non-epinephrine by adrenal medullae. But there's no parasympathetic effects. So yes, there's just different. I really do like this graph though. Yes. But it's just a lot of similarities, a lot of differences. Blood vessels, the iris, bronchioles, digestive tract, urinary bladder. Yes. So like for the urinary system, the kidneys, sympathetic effects are going to be decreases urine production, while parasympathetic effects are going to be increases urine production. So see how these are like opposites of each other? While in the urinary bladder, sympathetic effects are going to be constricts internal sphincter and releases urinary bladder, while the parasympathetic are going to be like tenses urinary bladder and relaxes internal sphincter to eliminate urine. But yes, there's a lot of effects for each. And down here is just going to be like a comparison of the somatic nervous system and the autonomic nervous system. So when it comes to the somatic nervous system, upper motor neurons, there's going to include the upper motor neurons and the primary motor cortex, while for the autonomic nervous system, the visceral motor nuclei and the hypothalamus are going to be included. So for the SNS is going to be the somatic motor of the nuclei brainstem, while in the ANS is going to be more of like the paranglionic neurons. Let's see right here, like in the SNS, it's going to include like the skeletal muscles in both parts, while in more the ANS, the visceral effectors are going to be more like smooth muscle and glands and cardiac muscle and adipocytes. So yes. Yes. No. Yes, but this was a very fun unit. This was a very fun, I think, PowerPoint slide to do. It was one of my favorites. I loved using all the diagrams, and I will see you guys next week. Thank you for listening.